Hello, in this session today we will be discussing NEET MDS Biochemistry MCQs. So let's start. First question is, which glycogen storage disease doesn't affect muscles? So here the answer is type 1. Glycogen storage disease are group of inherited disorders that are characterized by deposition of abnormal type or quantity of glycogen in the tissue. They are mainly due to deficiency of one of the enzyme of glycogenolysis or glycogenesis. Different types of glycogen storage disease are type 1, that is von Gilkes disease. This is due to deficiency of glucose 6 phosphatase in liver and kidney, and is the commonest type. So in type 1 there is enlargement of liver and kidney, hypoglycemia is associated, ketosis and hyperlipidemia. Second is type 2 is Pompe's disease, that is due to deficiency of acid maltase. Here glycogen accumulation in lysosome. Type 3 is Coleridge disease, that is deficiency of branching enzyme in liver, muscles and heart. There is swollen abdomen, growth is delayed, and there is low blood sugar with increased fat. Type 4 is amylopectinosis, that is Anderson's disease. Here there is absence of branching enzyme. Glycogen formed have no or few branches. There is progressive cirrhosis of the liver. Type 5 is McArdle's disease. This is due to muscle glycogen phosphorylase. Glycogen is accumulated in muscles and breaking does not occur. Type 6 is Her's disease. This is due to deficiency of liver phosphorylase. There is liver enlargement, low blood sugar. Type 7 is Tarwe's disease, deficiency of phosphofructokinase enzyme in the muscle. There are muscle cramps and anemia. Type 8 is associated with muscle weakness and anemia. Next question is, gluconeogenesis in fasting state is indicated by? It is indicated by pyruvate carboxylase activation by acetyl-CoA. So in gluconeogenesis, during short-term fasting period, liver produces and releases glucose, that is through glycogenolysis. And during prolonged fasting, glycogen is depleted and hepatocytes synthesize glucose through gluconeogenesis, and they use lactate, pyruvate, glycerol and amino acids. Pep carboxykinase, that is phosphoenol pyruvate carboxykinase, catalyzes the rate limiting reaction in gluconeogenesis. Next question, which of the following substances act to increase the release of calcium from endoplasmic reticulum? Here the answer is inositol triphosphate. So this IP3 or inositol triphosphate and diacylglycerol, they are second messengers for G protein coupled receptors. So IP3 stimulates release of calcium from endoplasmic reticulum and DAG activates protein kinase. Next question, phenylketonuria is disease due to improper metabolism of amino acid. So it is characterized by accumulation of phenylalanine. So this is basically mutation in the PAH gene, that is phenylalanine hydroxylase enzyme is affected. There is a defect in the liver enzyme, that is phenylalanine hydroxylase. There is impaired conversion of phenylalanine to tyrosine. Tyrosine deficiency, that leads to decreased neurotransmitters melanin and thyroxine synthesis. Next is, which of the following enzymes in gluconeogenesis bypass an irreversible reaction in glycolysis? So here the answer is 6-phosphofructokinase. The pathway uses several enzymes of glycolysis with the exception of enzymes of irreversible step, namely pyruvate kinase, 6-phosphofructokinase and hexokinase. Pep carboxykinase is the enzyme that is used in gluconeogenesis but not in glycolysis, as it converts oxaloacetate into phosphoenol pyruvate. So moving to next question. The principal store of metabolic energy available during long-term fasting of starvation is? Here the answer is adipose tissue triacylglycerol. Liver generated glucose and ketone bodies provide essential metabolic fuel for extrahepatic tissue during starvation and exercise. Next question is, which of the following metabolic alterations would most likely be present in chronic alcoholic compared to a non-drinker? So here in this case the answer is the ratio of NADH to NAD positive is increased. So in this case chronic alcohol consumption decreases acetaldehyde oxidation. Acetaldehyde generation is increased by chronic alcohol consumption because of metabolic adaptation. Next question is, all are actions of insulin except. So here the answer is gluconeogenesis. Here insulin inhibits gluconeogenesis. Insulin promotes uptake of glucose from blood and drive it inside the cells for utilization of energy production, that is glycolysis, storage in the form of glycogen, that is glycogenesis, and storage as lipids, so lipogenesis. That's all for today. Watch more videos on AdjuInfy. for NEET MDS. Do share, subscribe and like for more updates. Thanks for watching.